So in the previous video we were looking at transcription, and the final step of transcription is just simply termination. Basically, we've got DNA that has served as a template for creating this single strand RNA, again in the 5' to 3' direction. And once that RNA has been completed, notice the DNA zips back up and that single-stranded RNA comes off. And so again, the way it knows it's completed is it's going to hit a terminator sequence, and that's how it knows the RNA has reached the end of the gene. Okay. And so this next step, remember, is one of the things that's special and unique about eukaryotes is they do an extra little step called RNA processing. And RNA processing really is twofold. Okay, so there's basically two steps that happens. There's a capping step and a splicing step. Okay, and so the capping step, really the purpose is it helps the RNA because it is going to get exported, not out of the cell, but outside of the nucleus. It's going to be exported from the nucleus. Inside the cytoplasm, there's of course going to be enzymes and various things that could digest our newly made RNA, and so that's why the capping step is going to protect that RNA. And then also it's going to facilitate ribosome attachment. So the ribosome is going to be able to recognize which end is which and know where to start. So specifically the 5' end, which is kind of the end it's going to start with, is going to simply receive a G, a guanine cap. Okay. The 3' end gets adenine, but it's not just a single adenine, it's a series of adenines. It could really be several hundred adenines. And so they usually just call this one a poly-A tail because there are multiple adenines. Okay, so a poly-A tail. Okay, so down here in this picture, that's basically, imagine if this is the 5' end. Imagine a guanine cap, and then the 3' end, imagine a series of adenines, and so on. So hundreds maybe. Okay, and so that's the capping step. And now we're going to look at splicing, and there's still some mystery about splicing as far as really why it happens. But basically what the idea is there are some intervening sequences. Okay, we call them introns. So think of introns meaning they're intervening, and so we want them out of the way. Okay, so they're actually going to get removed. So in this example here, you'll notice there's an intervening sequence here and an intervening sequence here. And so basically they are spliced out. So just imagine a structure called a spliceosome coming in and cutting out those sequences. And then what that means then is this exon and this exon and this exon then get joined together to create this. Okay, so a lot of times it's kind of confusing because you think exon, they must be taken out. But really they're called exons because they will be expressed, meaning they will be used to produce protein. Or you can think about it as exons are going to exit the nucleus, go find a ribosome and make some protein. Okay, so there's definitely alternate ways that things can be spliced. So, maybe at a different point in our lifetime, maybe this is an intron here, and then this is the exon. And so obviously you can actually create a variety of polypeptides from one gene. Okay. So this is translation just as an overview. And so we've called translation other things before. So when we were studying the ribosome, we called translation protein synthesis. And so that happens at ribosomes. It could be free ribosomes, they could be bound ribosomes. And then the other review really on here is knowing that these amino acids, remember, are the building blocks of the proteins. Okay, so just remember that amino acids are the building blocks of proteins. Okay. So we're going to look at this a little closely. So knowing that amino acids are the building blocks, we need an RNA that's going to serve the function of being kind of a delivery truck. Okay, so we've got another type of RNA called transfer RNA, or just simply tRNA. And tRNA is going to not only carry an amino acid, sorry about that, got an email coming in, but it also is going to have something called an anticodon. Okay, we've seen codon before. Anytime you see codon, you should be thinking mRNA. So when you see anticodon, you should be thinking tRNA. So let me show you what that structure is going to look like. Okay, so here's the tRNA. Again, it's an RNA, so if you look down through here, you'll see A's and C's and G's and U's. Okay, no T's in there. But again, on one end, this 3' end, this is where the amino acid would attach, right here. So imagine maybe if it's the, MET or something. Okay, any kind of amino acid could connect there. And then on the opposite end over here, okay, is going to be an anticodon. So again, it's a group of 3 letters, a triplet, and really the first 2 are going to be complementary to the codon. So again, if there's A's here, this would be U, U. Okay, it could be C, because C goes with G. But again, that third letter in the anticodon does not have to be perfectly. So it does kind of wobble in place because of that third one doesn't have to be a perfect match. Okay, so anytime you see this symbol, this is the symbol that they're going to use for the tRNA. Okay. So again, we have 3 stages of translation, just like we had 3 stages of transcription. So same names, initiation, elongation, and termination. This does require energy. So remember earlier in the semester we talked about anabolism. Anytime you're building something, and this is an endergonic reaction, meaning it requires an energy input. Okay, usually in the form of ATP. Sometimes we'll see GTP. So instead of adenine, it's a guanine. Okay, so you can see that right here. So anytime you see that starburst, know that that step requires energy. Okay, so this very first step, this is initiation. So this is the first step. And you will see in initiation, there's several things happening. Okay, so you'll see there's 2 subunits, 2 parts of a ribosome. There's this smaller bottom one and then this larger one here on top. So notice the messenger RNA here in red. So messenger RNA is going to basically be sandwiched in between those 2 building blocks of the ribosome. Okay, and then if we zoom in and take a look at this site right here, you will see, sure enough, AUG is our start codon. And if you look back at your genetic code earlier in the PowerPoint or open your book to that, you will see AUG codes for the amino acid methionine. Okay, so MET will be the amino acid that tRNA is delivering. Again, remember if you looked at this anticodon right here, it would be complementary, at least in those first 2 locations. So we'll go ahead and just assume it's complementary for all 3. So UAC would be the anticodon that matches AUG, which is the codon. Okay, so again, imagine we have methionine or MET binding to that location. Okay, so that's initiation. And if we zoomed forward and looked at this, your book doesn't have a great picture of this, so I'm going to go ahead and insert one. So imagine, again, we would have this ribosome that has a large and a small subunit, and our messenger RNA that's woven through there. Okay, and so then we have we'll talk about these 3 locations on the ribosome. Okay, so if you look back at that last picture, the first one during initiation, is going to actually be here in this P site, okay, again with methionine attached. And then a second tRNA arrives carrying a second amino acid, so I'll just write down amino acid number 2. Okay, and if we look at this, even the very next step, you would see methionine is actually going to hop over here and a peptide bond, remember a peptide bond connects amino acids, peptide bond forms between that first amino acid methionine and that second one that's brought in. Okay. Again, if we look at it, the very next step, again, if I just kind of zoom into that ribosome, okay, and we have these 3 sites. Okay, if we fast forward one little still step, you would see the tRNA that was here in this A site has slid over into the P site, and it now has the amino acid number 2 with bonded with a peptide bond to methionine. Okay, the initial tRNA that brought in methionine is slid over here into this site, and notice it no longer has that amino acid anymore. And then we also would have a third tRNA that has arrived at the A site with a third amino acid. Okay, and then imagine these here coming over and forming a peptide bond and linking together. So we're basically making our little chain of amino acids. Okay, so your book kind of skips over a few of those steps. So if we kind of catch up, this has basically it has 4 amino acids attached here, and then we have number 5 arriving here into this site. Okay, again, our chain of 4, this little red arrow represents how that's going to move over and form a peptide bond with amino acid number 5. And you can kind of follow that. So now all of this is just basically going to slide over. Okay, so when we get to this picture here, I can add some arrows in here. So this empty one is going to slide this way. This one in the A site is going to slide over into the P site. And so then that brings us to what these E, P, and A stand for. Okay, so the best way to think about it is if you notice what happens to the tRNA in the E site, it exits. Okay, so you can think of E for exiting. So that tRNA would actually be going and picking up an amino acid and possibly coming back around. So it just gets recycled. Okay, if you look at the P site, okay, most of the time that P site is actually holding the growing polypeptide. Okay, so you can think of P for polypeptide. The only exception to that is really during initiation. Remember, our very, very first amino acid, methionine, was connected to a tRNA that was in that P site. Okay, but most of the time that's where that growing polypeptide chain is going to be held. And then lastly, the A, think amino acid. Okay, think single amino acid or think arrival, because that's where that first, each single amino acid is going to arrive there. Again, with the exception of that initial methionine. Okay. The last part of this is termination. Again, here's where we have our 3 stop codons. Okay, if you look back again at your genetic code, we have 3 codons that do not code for an amino acid. So when one of those stop codons arrives, instead of tRNA, we have a release factor that binds, and it does exactly that. Okay, so if you advance forward a couple slides, you'll see everything is released. Okay, especially, most importantly, our completed chain of amino acids is released. Okay, so I'm going to go ahead and stop this video, go take a break. You will need a piece of scratch paper, piece of paper that you're going to actually turn in to me on Monday.